So today's lecture on Biochem 510, that is carbon and nitrogen metabolism. In this lecture we will understand the synthesis and role of trehalose. So trehalose is a quantitatively important compatible solute and it is stress protectant in many organisms. So trehalose is important. Why? Because it works as a stress protectant in many organisms, including which are the organism comes under and what is their role, trehalose in that. So primitive plants, including any green plants or green synthesis or chloroplast synthesis or chlorophyll synthesis are involved in that. So that is related with stress protectant and compatible solutes. These functions I am telling about compatible solute have largely been replaced by sucrose in vascular plants. Trehalose metabolism have taken a new roles too, which are that so it can also have a potential to work as a signal metabolites in many plant interactions with that may be a pathogenic one or maybe a symbiotic microorganism or it can also work in herbivorous insect. So generally it will comes in microorganisms and herbivorous insects are involved or work as a signal molecules or signal metabolites. Another functions it is implicated in response to cold and stress or cold and salinity. Why and how? That in regulation of stomatal conduction and water use efficiency. Now, the chemical formulas of trehalose, how it's look like. So it is, you know very well, it is a non-reducing disaccharide formed through 1,1 alpha bonds with linkage of two gluconopyranosides. So you can say alpha D gluconopyranosyl 1 1 D alpha D gluconopyranoside. These two join together with which bond? 1,1 alpha bond. Okay. So variety of organisms synthesize these compounds, including plant, fungi, bacteria, and invertebrates animals too. In many books, you will find only an insect. Wherever you are studying trehalose, they will tell you that it is a main blood sugar which is found in the insect. Okay, so red one you have to remember, and trehalose is the main blood sugar in the insect, and serve as a major storage energy molecules enabling flight. So one can ask you why insect can fly. What is the chemistry behind that and what chemical is responsible for that? So you can say the energy comes from sugars and it is a disaccharide, non-reducing sugar, and it is found in the blood sugars in the insect. Therefore, it serves as a main storage and it can fly. Now, trehalose in the plant. How? In those plants that accumulate trehalose, it is mostly commonly believed to or it is have a ability to extend period of desiccation. During the process of desiccation or defoliations or however few exceptions, this is unlikely to stem from direct role of trehalose itself since only a trace amount of trehalose are present in blanks plants. So you have to write angiosperm. There are numerous research has been detected a role of trehalose. questions comes in your minds or my minds also. How it is synthesized, what is the precursor, what metabolism is occurred. So Trello's precursor is Trello 6 phosphate, TC, T6P, a regulatory molecules that is especially in the sugar influx and the metabolism. There are a multiple biosynthetic pathways are also available. You can ask that, sir, how it is possible? But according to the species, whether it is in bacteria or whether it is in archaea, but also one has found in eukaryotic and examined certain data also. And to date, the scientists is also effectively found the eukaryotic pathway shares many similarities as you found in the sucrose synthesis in the plant. So Trellose is synthesized. Now metabolism. Remember this sentence. Trellose is synthesized via phosphorylated intermediates. As I said previously in the previous sentence, Trellose 6 phosphate. A meta-analysis of revealed that the Trellose 6 phosphate change in parallel with the sucrose. So which is the major product of photosynthesis? Sugar. We know very well. And the main transport sugar in the plant, sucrose. So first step in the UDP glucose and glucose 6 phosphate linked to or linked by T6 synthase is the enzyme form Trellose 6 phosphate. So first step is what? UDP glucose plus glucose 6 phosphate that is linked by Trellose 6 phosphate synthase is the enzyme and to form Trellose 6 phosphate. So phosphate group is removed by T65, T6P, TPP, phosphatase is the enzyme. And finally you will find Trellose 6 phosphate to Trellose. So Trellose is a subsequently broken down into a two molecules of glucose by Trellase. Enzyme is Trellase. So in this para you will find a three enzyme. One is phosphatase, another is Trellase, and third is synthase. Okay. So the three enzyme you have to remember for Trellose biosynthesis. Now existence of bidirectional network in Trellose 6 phosphate is a signal of sucrose availability, and it also acts to maintain a sucrose concentration within the appropriate range. Now relative amount of this sucrose Trellose 6 phosphate influence, okay, and starch that accumulate in leaves during the day. Starch is accumulated in the leaves during the daytime, and its regulation of or rate of starch regulation or starch degradation at night to match the demand of sucrose. Mutant, for example, T Trellose 6 phosphate metabolism have dryly, directly, or you have a highly pleiotropic phenotypes show defect in embryogenesis, leaf growth, flowering, inflorescence branching, and seed set. So these pathways is now evaluated. In many books, you will not find Trellose 6 phosphate is found in the plant. They are generally known only insect blood sugar. Okay, so it has been proposed that Trellose 6 phosphate influences the plant growth, development via inhibition of this SNF and SNRK1 that related protein kinase, or you can say that SNF1 related protein is regulated. Now the regulation of the starch synthesis by Trellose 6 phosphate. So As you know that in carbon metabolism of the plant acquire carbon via photosynthesis, and this is only occur in the presence of light. You know very well. During the daytime, part of phosphatate is used for the synthesis. At night, export of sucrose to support growth of zinc organ. Most of the remainder is accumulated as a starch in the chloroplast, and starch is remobilized at night and used to support respiratory metabolism. Remember this sentence: starch is remobilized at night and used to support respiratory metabolism in the liquid, or to synthesize sucrose. These two words. What is the function of starch during night in the plant? Starch is remobilized at night and used to support respiratory metabolism in the leaf, and or to synthesize sucrose, and that is exported to support growth of zinc during the night. This transitory starch reserves enable the plant to cope with the daily challenges of surviving through the night, and must be prudently managed to optimize growth rates and achieving reproductive success. So now the question for you people: what is the effect of Trehalose 6 phosphate? I have already told you here. It has a pleiotropic phenotypic effect. It also affect if it is not found in proper amount or proper balancing of sucrose synthesis. It defects in the embryogenesis. It also involved in the leaf growth. It is also involved in the to support the respiratory metabolism in the leaf, or it also involved in the seed cell. It is also involved in the inflorescence or branching of the leaf tissues in the plant. So this is all about the regulation of starch synthesis and its effect in the trehalose, main blood sugar, how it is synthesized, and what is their functions. This is all comes under the oligosaccharides. Okay, so this is